Hi everyone, today I'm going to be reviewing the paper, The Genotype-Phenotype Correlations of the CACNA1A-related Neurodevelopmental Disorders, a small case series in literature reviews. This article was published by Kessi et al. In the journal Frontiers in Molecular Neuroscience in 2023. So I know the title of the paper sounds complicated, but let's break it down. First, what is CACNA1A? CACNA1A is a gene that codes for the Cav2.1 channel, which allows calcium to enter neurons. This is important for neurotransmission, which is how neurons in the brain communicate with each other. Here are some important terms to know. First, I want to define what a variant is. A variant is a permanent change in the DNA sequence of a gene. It could be a single nucleotide change, or part of the DNA sequence could be missing. A gain-of-function variant is a change in the DNA sequence that increases the amount of calcium going into neurons, perhaps by opening too much. A loss-of-function variant is the opposite. It decreases the amount of calcium ions going in, perhaps by not opening as often. A missense variant is when a single change in the DNA sequence leads to a change in the protein sequence. CACNA1A variants can lead to a wide range of symptoms or phenotypes. The ones that the researchers focused on in this study were epilepsy, global developmental delay and intellectual disability, which I will be referring to as GDD and ID from now on, and autism spectrum disorder. The reason that the authors focused on these phenotypes was that they were looking for a connection between the type of variant or genotype and its phenotype. To answer this question, the researchers pulled data from published literature about patient variants. They also analyzed data from 6 patients in a hospital cohort. For more information on this cohort and the variants included, please see the paper, which I will have linked at the end of this presentation. First, let's review their results on epilepsy. They found that the majority of patients with epilepsy had the missense type of variants as opposed to a nonsense or deletion, where the protein could be gone or parts could be missing. They also found that, although the majority of patient data did not include the variant's functional impact, that loss-of-function variants were the most common. While there were more loss-of-function variants linked to epilepsy than gain-of-function, the 3 most recurrent individual variants for epilepsy were A713T, V1392M, and V1393M, which are gain-of-function variants. The researchers also looked into specific types of epilepsy. One type is status epilepticus, which is defined by seizures lasting longer than 5 minutes or without a break in between. Another type of seizure they analyzed were absent seizures, which result in sudden lapses of attention or consciousness. They found that status epilepticus was more common in gain-of-function variants, and that absent seizures were much more common in loss-of-function variants. The researchers also looked at the effects of anti-seizure medications, or ASMs, on CACNA1A-related seizures. They defined seizures as either controlled by medication or refractory, which are seizures that don't respond to an ASM. They found that over 50% of epilepsy patients had seizures that didn't respond to medications. This brings into question the effectiveness of ASMs for CACNA1A-related epilepsy. Next, the researchers looked at GDD and ID. They found that missense variants were once again the most common type of variant to cause GDD and ID. Looking at the functional impact of variants, they found that the majority of the known variants were loss of function. As with epilepsy, the most recurrent individual variants included the gain-of-function A713T and V1393M variants. Another pattern that the researchers noticed was that gain-of-function variants showed severe GDD and ID, while loss-of-function variants were milder. This graphic reiterates the pattern found in the last slide, this time looking at the severity of GDD and ID in recurrent variants. For example, you see more orange, which represents severe GDD and ID in mainly gain-of-function variants. You see more blue, which represents mild to moderate GDD and ID in mainly loss-of-function variants. Moving on to ASD, the researchers found that the majority of variants associated with ASD were missense variants. Gain of function was the most prevalent functional impact, which contrasts the common functional impacts for the other two phenotypes. The most recurrent variant for ASD was the V1392M variant, which was recurrent in all three phenotypes. To summarize, the researchers found that loss-of-function variants are associated with absence seizures and mild to moderate GDD and ID. Gain-of-function variants, on the other hand, are associated with status epilepticus and severe to profound GDD and ID. Finally, they found that refractory epilepsy is present in over 50% of epilepsy patients, signaling that more effective anti-seizure medications are needed for CACNA1A patients. Thank you for listening.